Everyone, thank you so much for coming today. This is a CANSSI Ontario Statistics Seminar, part of the CAST program. I am associated with that in that I am the local person who manages CANSSI for Trent and encouraged our joining of CANSSI. That's the Canadian Statistical Sciences Institute. So it's a cross-country group, kind of like Fields is for math, to try and promote statistics research. And these talks are supposed to spotlight groundbreaking research in statistical sciences, both applied and theoretical. And for your benefit, we went with applied today. And the goal here is just that participants can engage with leading experts from across Canada and even around the world. And because we're going to stream it, and we are streaming it hybrid, people can watch it from all across the country. So there may be people tuning in from BC or the Maritimes. There's about 60 or 70 people who are going to watch it online to be part of this talk. So it's not just people at Trent, it's anybody who wants to come along. And so this seminar is a collaboration between Trent University, the ENLS program, CEMC, and the Department of Math and Stats, just to tag a bunch of things, and our guest, Dr. Ben Bolker, who's here from McMaster University. Ben did his PhD in the UK at Cambridge, postdoc at Princeton, and was in Florida. And then we stole him and we brought him up to Canada. And then he's been here since 2010, I think. And Ben is one of those very strange ducks. He is both a genuinely trained zoologist of sorts, biologist of sorts.
I have a PhD in zoology.
There you go. And a statistician. And he's cross-appointed and is in a Department of Math and Stats and does genuine statistics research while simultaneously knowing the field very well at the level that people like me could never hope to, because we just narrowly trained in stats. And so he's worked on all kinds of things. If you check his CV, you'll see papers across the breadth of ecology and biology and things that happened to spark his interest when he was bored one day. And today he's going to be talking to you all about fish, but also genetics and MCMC and stats. And so I think that's all I want to say. I'm going to pass it over to Ben.
That was great. That was not unduly embarrassing. So I can't ask the people online, but I just want to, if you would mainly identify as a biologist, please raise your hand. Okay, if you would mainly identify as a statistician, please raise your hand. Okay, that's information. This is a stats seminar because I was invited, but we'll get there together. The other thing is, please do interrupt, ask for clarification, point out typos on the slides. This is a problem in figuring out the influence of behavioral traits on the evolution of morphological traits. And so the statistics part is, how do we model and estimate the interdependence of evolutionary rates, or the dependence of rates of evolution of some traits on the states of other traits. And I will tell you more about these traits in a moment. I always run out of time and rush through my acknowledgments and basically money. And student Sigal Balschein's a colleague of mine at McMaster. Her students, and there's more than are listed here, spent a huge amount of time doing a literature survey, a meta-analysis data collection that I'll tell you about. The evolutionary, so the evolution, or really the statistical evolutionary problem, is we're interested in the correlations. We're interested in how do these different traits go together. But these traits live on a phylogenetic tree. We're measuring the traits of extant organisms. We know a lot about their relationships. And so we can't just do regressions, because we have to account for the fact that the members of a family or a clade or a genus are related and are likely, there's a whole conversation here, but they're likely to have similar traits just because they're evolved from the same, descended from the same ancestor. And so we want to deal with that appropriately. And the specific, this is a big field. This is the field of phylogenetic comparative analysis. But I'm specifically talking today about the evolution of traits that we're going to treat as discrete. I want to talk about the evolution of accessory glands. What the heck is an accessory gland? It's an organ involved in reproduction, but not directly in the production or release of gametes. Typically, these are some kind of glands that are putting stuff into that are secreting stuff and putting it into seminal fluid, like I just said. And they're often associated in lots of organisms where there's some kind of open spawning, where a female can have multiple male mates. There's often sperm competition. So there's competition going on at the chemical level between sperm from multiple males about who gets the paternity from that female's eggs. The interesting part, and the reason Segal-Balshine came to me, is that many fish have a significant component of male parental care. The males are fanning the eggs to make sure they get enough oxygen. They're defending them from predators. They are also defending them from threats like the overgrowth of fungi. So there may very well be, we don't know much about fish seminal fluid, but there may well be stuff in fish seminal fluid that's helping the eggs survive and develop properly. And so what we want to try to test is, can the evolutionary data, can the distribution of these traits in extant organisms tell us something about why this trait evolved? Not that many fish have accessory glands. These are examples of some of the families that where these, especially lots in catfish, and you'll see a phylogeny in a moment, but these are some of the families of fish that do have accessory glands. This is the huge amount of work that the students did. They went to the literature and they found every fish species where somebody had looked at the reproductive anatomy, the male reproductive anatomy, carefully enough to say whether they did or didn't have accessory glands. So either they explicitly said they didn't have accessory glands, or you could see from the amount of detail that they had on the morphology that if they had accessory glands, you would have seen them. So as I said, there aren't, and they found this information on 607 species. These are all ray-finned fishes. And it's pretty concentrated in a few families. 26 families, 116 species. And if you do a basic sort of parsimony analysis of how many times do we think this trait has been gained or lost since the ancestor of the ray-finned fishes, or at least of these 607 species, it's been gained about 20 times and lost about 9 times. And there's uncertainty associated with that. The other thing they collected when they could find it, so for species where we don't know anything about the accessory glands, there's not actually that much point in trying to get this data, but also whether we think there's sperm competition going on. And so that's essentially, is spawning events or mating events going on just between a single male and female at a given time? Or is there some kind of broadcast spawning? Or is there a whole group that's present at a mating event? And whether there's male parental care. In the full data, we know about whether there's female only, biparental, or male. We're lumping biparental and male together in this case, because in both of those cases, the male is caring for the eggs. And so this is that phylogeny again with names that you can't possibly read. And then in addition to the information about, sorry, I realize people are seeing the video from there, so they can see me pointing at the screen. I would hope somebody would have stopped me before now if that weren't true. We also have information in this ring about whether the spawning mode of this particular species, whether this particular species does group spawning or pair spawning, and whether they have male care or not. One thing you'll notice is there's a lot of white space in these rings, and those are missing data. Because just because somebody dissected the reproductive anatomy doesn't mean that they've watched the fish mate or that they know anything about parental care. So this is the data we've got, and we're going to try to put this together and build a statistical model to figure out what's going on. This is sort of the, I'll talk briefly about consensus trees and tree blocks in a minute. But this is the basic breakdown of we know everything of these 607 species. We only have both of these, we only know both of these traits for 261 of them, and the others are distributed in these. Maybe we know one or the other, or maybe we don't know anything. So that's the evolutionary story, and I'm open to questions about the biology, although any really deep questions about the biology I won't be able to answer. People have been interested in this problem. How do we estimate the rates of evolution of discrete characters down a phylogeny? So we imagine that as we go along the branches of a phylogeny, that these traits are flipping on and off. They're discrete traits, so at particular moments in evolutionary history, they're turning on and off. This red is our sort of a good guess of the turning on and off of the accessory glands. But we're going to want to look at a multi-state system where all three of these traits are turning on and off over time. Although since we're assuming that evolution works in continuous time, we're going to assume that only one of them switches at any given moment. As I said, this is a question that people have had for a long time. Mark Pagel is kind of the grandfather of this. And so the statistical question we're asking is the probability that accessory glands are gained or lost along an evolutionary lineage independent of the states of the other two traits, independent of whether that lineage at that moment in evolutionary history has male parental care or doesn't, or has group or pair spawning. And what Pagel liked to do, and there's going to be some statistical philosophy in here as well, which is either good news or bad news, depending on how much you like statistical philosophy. What Pagel et al. Like to do is do a bunch of hypothesis tests. This is just a screenshot from one of their papers about whether we can reject, basically, can we reject the null hypothesis that the change in a trait doesn't depend on the state of X? So can we say it does depend because we were able to reject that particular null hypothesis? And this tends to, there are a lot, I'll show you, there's a lot of parameters involved here. There's a lot of rates, and we end up doing a lot of comparisons of different rates, and that's a thing I want to get away from. And these are likelihood ratio tests for those for whom that matters. It's a continuous time discrete state Markov process. Markov process just means that what's happening in the system only depends on the immediate history. I don't have to look back in the phylogeny to see what were the great-grandparents of these things doing. And I'm going to define the rates. Probabilities in continuous time are a little bit weird because you're looking, the probability that something happens in an infinitesimal moment of time is 0. So you end up taking, end up getting basically, oh, there's a missing divided by delta t here, so that's good. So I found my own typo. This is essentially, you can think about this as the instantaneous probability or the instantaneous rate of transition between state I and j. I have 3 traits, each of which I'm treating as binary. So I'm going to unpack this and there's 8 possible states that the evolutionary trajectory could be in, which are all the combinations of spawning mode, parental care, and presence or absence of accessory glands. So I'm going to be looking at an 8x8 transition matrix, the probabilities of going between all of those states. So each of these little bubbles is whether accessory glands are present or absent, whether parental care, male parental care is present or absent, and whether the spawning mode is group. And these red arrows are where the AG bubbles turn from white to gray. These are the transitions that I'm especially interested in. And I want to see, and the black arrows, the dotted arrows, I can't remember whether gray is present and white is absent or the other way around, doesn't matter. Arrows going in one direction are gains, arrows going in the other direction are losses. And I'm interested in the red arrows because those are the rates of gain and loss of accessory glands. depending on which of these corners I'm starting from, depending on what the other two states are doing at that time. I'm going to make a big simplification, which is to assume that the other traits, that parental care and spawning mode, are changing back and forth at a rate that's independent of each other and whether there are accessory glands. So I'm really interested in all 8 of these arrows. And many of these arrows I'm just going to assume are the same. The rate of gaining or losing parental care here is the same as the rate of gaining and losing parental care here. And I may come back to that. And this is either, yes, no, Sigal, I can't talk to you right now. I'm talking about your work right now. So I've got a model, at least I've got a conceptual model. There's a mathematical statistical model underneath it. I'm going to make an 8 by 8 matrix that is the rates of each of those transitions. Some of those transitions aren't allowed. You'll notice here, I don't have arrows. I don't have a whole bunch of diagonal arrows. And that's because I'm in continuous time. I'm assuming only one thing, only one of these subtraits changes at a time. So luckily, when I write down this matrix, there's going to be a lot of zeros in it, which is kind of going to save me. And for mathematical or technical reasons, I write down the probability of changing from yourself to yourself as I want to make sure that the rows sum to 0 for technical reasons. And there's some kind of cool math when I look at a branch, when I go from branch of a phylogeny. I'll get back to a phylogeny eventually. When I say, well, if I know the state here, what happened, what changes, if I know the probabilities of being in all states here, what changes as I go from here to here? And I have to take into account that along that continuous time, if the rates are fast enough, there might have been a lot of changes through intermediate steps that I can't possibly see. Again, cool math that I'm not going to talk about, but it turns out that there's a matrix exponential, which if I have the transition rate matrix, this delta t is the branch length in this phylogeny. This is how much time was there for evolution to happen. And then the q are the instantaneous rates. And this exponential, if I have gives me the probability of going from one state at the beginning of the branch to some other state at the end of the branch. OK, there's one more hard part here. So this gives me a probability. If I know where I was at the beginning of a branch, this tells me what the probability is of moving to another state by the end of that branch. But I've got a whole tree here, and the only thing I know are the only data I have are the values at the tips. So I somehow have to compute a probability over all of the possible evolutionary trajectories of over all of the branches in this tree. It turns out that there's one more kind of cool mathematical or algorithmic trick for figuring this out. And this goes all the way back to, so if Pagel is, I don't know, Pagel's the grandfather, Felsenstein is the great-grandfather. Felsenstein is another very, very smart, very big person. Probably, I would say, started the field of phylogenetic comparative analysis. Basically, this is an algorithm for computing given parameters for the rates of all of these transitions. This is a way to calculate the likelihood, the probability of ending up with the tip data that you actually see. And what it does basically is start from the tips and go backwards and work at the tips. You know what the probabilities of those states are. At the tips, I've observed that species. So I've got a 1 for this combination of traits and a 0 for everything else. And what I can do is work my way backwards and say, if I know what's happening at this set of children, I can go back and calculate the probability at their common ancestor. And I have a quick little demonstration here. So here, this is a 3-state model. I'm not going to show you an 8-state model, where this species is in state 1. This species, I've labeled these 0, 1, 2. This one is in state 0. This one is in state 1. So there's a 0 for these two and a 1 for the middle, and so forth. And that's what I know. I know the probabilities that these species that I can measure are in a particular state. And what the pruning algorithm does is if I know these two, I can work backwards and calculate this probability. And then I can just go through the tree. And now I know this one. And so I can go back and calculate this node. And now I'm going to calculate this node from the two that I think I know now. And then I can go back and calculate. And when I finish, I do have to have a prior probability. I have to have some reasonable guess or distribution about what's going on at the root of that tree in order to finish my calculation. This is fast, it's convenient, it's kind of magical. Now I have, for a set of parameters that are describing the rates, I have an algorithm that can very quickly give me the likelihood, which means I can do maximum likelihood estimation, which means I can estimate these parameters and get confidence intervals and test hypotheses. There's a package that was around before I got there. Core HMM is correlated hidden Markov model. Independent is my hypothesis that gaining and losing accessory glands doesn't depend on the states of either on sperm competition or spawning mode or parental care. PCSC is an interaction model. It says it depends on both of them, and it depends on them differently depending on what the other one is. This is an additive model. It depends only on parental care, but it's independent of sperm competition or spawning mode and vice versa. So I can do the way that likelihood ratio tests work. I can do a hypothesis test for any of these particular comparisons. And this is a very standard statistical approach, and I find it kind of frustrating because, well, do I want to test? Oh, I didn't draw a line. I could test independence versus the interaction model as well. There could be a curvy arrow here. I can draw anyway. And there's little p-values here that are too small for you to see. What I don't like about this is that this gives me a bunch of individual hypotheses tests, but I don't feel like it gives me, okay, how important is this? This just tells me, can I see this or not? So here's the pause for statistical philosophy. I'm going to see how I'm doing on time, which is not great. So I find these pairwise comparisons unsatisfying. I'm always yelling at my students, don't just tell me whether it's significant or not. Tell me how big the difference is. How uncertain am I about the difference? Is that difference biologically important or not? So this is the kind of conclusion that I might get out of assembling a bunch of hypotheses tests. Well, the interaction term and the combined effect are non-significant, but everything else is significant. I don't like it. Another thing I could do that people do very often in wildlife biology, conservation biology, is they'll fit all the different models, and they'll compute something like an AIC score or a BIC score and say, which model predicts this best? And in this particular case, the one that wins is accessory glands are dependent on, this is the model that says that parental care and spawning mode both affect the evolution, but they don't interact with each other. This still doesn't give me magnitudes. This still says which of these discrete hypotheses is better. Furthermore, AIC at least was designed for prediction. It wasn't designed for inference. And so here I am trying to make an inference, and it makes me nervous. So what about a Bayesian solution? I need to make things a little bit, that's too much smaller. I'll make it small for a moment, and then I'll come back. I'm not the first person to think that it would be a good idea to go to a Bayesian model where I could integrate over all the different sources of uncertainty, but I'm going to be snarky about the way that these people chose to do it. The way Pagel et al. Did something super clever and, in my opinion, misguided. They said, we've got all these different rates. We've got all these different combinations of traits and rates. And what we're going to do as a hypothesis is say, well, what if rates 1, 2, and 1, 3 are the same? And this is just one of the many possible hypotheses you could generate. 1, 3, and 2, 4 are the same. And they also allow for a group all to be 0. So what if this transition is impossible as a hypothesis? When you do that kind of partitioning of these sets of rates, you end up, if I had 12 rates, I would have 4,213,000 possible individual hypotheses, because there are lots of ways to group 12 things into discrete sets. And you need to do some really fancy technical stuff to be able to sample over this space in a reasonable way. But here's what I don't like about this. The posterior distribution that you get, since you're jumping around between these rates, you're averaging over, maybe this rate is 0, or maybe it's large. And these are cases where maybe it's large or maybe it's very large. I don't really buy this. If I just think about biology, I don't really buy this as a sort of sensible probability distribution over what I think. I think maybe I would imagine that the rates could be, my best guess would be they would be medium sized, and then I could see that smeared out. And occasionally I see multiple modes, but I think what we've done here is induced a really weird posterior by making a particular technical solution. So I actually, and I'm going to skip over that for the moment. And I'm going to skip over that. OK. So what I want to do is go back to quantifying things and say, what's the difference between the rate at which this happens if I have parental care, male parental care, or if I don't have male parental care? And the way I'm going to do that is Wesley said he was happy to see the students seeing that. I'm going to set up a contrast matrix, which is going to say essentially, so the intercept here is going to be what's the average of the gain rates across all the states that I might be in. So summing up a quarter, 1/4 times each of those, and then adding them up. The next one, the main effect of care, is the difference between the average rate if I do have care and the average rate if I don't have care, and so forth for spawning. And then I can also write out an interaction model, which is actually hard to do in the Pagel et al. Framework. So I have a way to set up my model so that I'm estimating the differences that I'm interested in. I told you at the beginning, we have in this 8x8 matrix, I have 24 rates I have to estimate, assuming that spawning and parental care are happening at their own rates and not dependent on each other, gets me down to 12 parameters I have to estimate. This is going to be hard. You saw at the beginning, yes, I have 600 species, but I have missing data on more than half of them. And the fact that those traits are concentrated in particular parts of the phylogeny mean I'm not really looking at very many independent events. So I really do need to do everything that I can. One thing that people who are learning Bayesian statistics for the first time, or actually lots of people who do Bayesian statistics even as experts, is they try to sweep the priors under the rug. They say, well, let's just make a prior that's as uninformative as we can. I'm going to use priors that are neutral, meaning they're going to be centered at 0. The contrasts are going to be centered at 0, which means I don't have an a priori opinion about whether having male parental care increases, or I do actually have an opinion, but I'm going to leave that out. I'm going to be neutral about whether the effect of male parental care on the rate of acquisition of accessory glands is positive or negative. but I'm going to make it regularizing. So I'm not going to allow it to stretch off to infinity because I've got very little data. And if I allow crazy answers, then the machine is quite possibly going to give me crazy answers. And probably all of my sampling won't work very well either. The other technical detail is that I mentioned that of these 607 species, I only have full genetic data for about 400 of them. So there's about 200 of them that I can't place in the phylogeny with certainty. What Rybowski et al. 2018 did, the people we got these phylogenies from, is they said, well, if we don't have genetic data from a species, we're going to take the taxonomic information we have, what genus is it in, what family is it in, and we're going to put it somewhere in that clade that we know that we think it belongs in, and we're going to sample the branch lengths in a sensible way from the distribution of branch lengths in the tree. What that means is that rather than doing this with one tree, we have 100 sort of equally plausible trees that we are running this model on. So again, apologies to the biologists who walked in and Metropolis Hastings sampling with adaptive multivariate normal candidate distribution is just mysterious. What we're going to do is every time we take a new guess at the parameters and evaluate the goodness of fit and the prior probability of those parameters, we're switching trees every time. So we're essentially averaging over the distribution of unknown trees. What's great about that is it lets us use all 600 species instead of being stuck with the 400 species where we actually know where they're supposed to fall in the phylogeny. And I've now said I've said about half of what I intended to say. So these are informative priors. They are changing my answer, but they're neutral. What they're doing is ruling out crazy answers. And the way that I did this is I said, what's a reasonable number of transitions what's a reasonable rate? What's a reasonable number of transitions that could happen over all of the evolutionary change in this tree? So a hazard that gives me like 0.1 expected change over the whole tree is not very realistic because I couldn't possibly estimate that anyway. Because I would not see any evolutionary change. And a hazard, if I assume that these traits are changing 1,000 times along every individual branch, that's also very unrealistic. So I basically came up with a range of rates that I thought was reasonable, or a range of total evolutionary transitions that would be reasonable, and set my prior to allow that range. I also did something that turns out not to have been very important, but evolutionarily we think it's probably easier to lose a morphological structure than to gain a morphological structure, because losing a trait typically means, oh, I'll have one knockout mutation and I can't make that properly, or I don't use that and drift happens and it accumulates silencing mutations. Whereas if I have to evolve accessory glands from scratch, that's a bit harder. So we made up some priors on what we thought the asymmetry between gain and loss might be. The other thing that we did, which is part of the evangelism here, is we looked at what these priors made the evolution look like. We picked values from the prior and we said, what answers would we get from these priors if we had no data? And we made sure that those weren't, in fact, biased in some direction we didn't intend them to be, and those didn't, in fact, give rise to any unrealistic outcomes. This took a while. Well, whether you think this is a long time or not depends on what kinds of computational problems you do. It took about 8 hours, about half a day to a day on 8 cores on a reasonable workstation. And the standard MCMC diagnostics all looked good after that length of time. I think I also drew trace plots. These are individual, so these are my individual parameters, my individual rates, loss of sperm competition, loss of parental care, and then these are gains or losses of accessory glands in particular conditions. And this is looking at the bivariate posterior distributions. I'm basically looking at this to make sure that it seems like it makes sense. This is really the kind of bottom line of the paper. So if we look at, these are the contrasts. These are the effects of male care. How much does male care proportionally, the presence of male care in the lineage, increase or decrease the probability of gaining or losing accessory glands? So we were happy and a little bit surprised that male care actually increases, having males care for the offspring increases your rate of gaining accessory glands about tenfold with a very large uncertainty. These are the 95% credible intervals. So if we're hypothesis testing, we say we think there's at least a 95% probability that that's positive. And we could, since this is Bayesian, we could actually calculate that probability directly. It's very hard to say anything about loss. All of these bubbles are basically centered at 0. And that's because there aren't very many species with accessory glands. And those all happen, those seem to have evolved fairly recently. So there's just not much, there aren't many parts of the tree where you could be losing accessory glands, because you can only lose accessory glands if you've already got them. So we really have not very much information about what's happening with the loss. And somewhat to our surprise, at least over this data set, it looks like group spawning actually slightly lowers your probability of gaining accessory glands. We did some sensitivity analyses. I'm actually not going to talk about them very much because I've realized that I'm looking at a version of this talk that's about 14 hours old, and I fixed some things about 12 hours ago. So I'm going to tell you the take-home message. So I'll tell you, I won't show you I will skim very quickly past the picture, which is wrong. What did we do? Before I told you we made some simplifications to get down from 24 rate parameters to only 12. We tried doing this not without the fancy phylogenetic imputation, just with the species that we knew about. We dropped the gain and loss priors to see whether that made a difference. We compared this to the maximum likelihood estimates, because we can also get confidence intervals for those. And we compared those. MAP is maximum a posteriori. So this is like, what if you're sort of halfway to being a Bayesian and you put a prior on something, but instead of doing the Bayesian sampling thing, you just look for the most likely set of parameters. And again, without letting you look at that for very long, if I do the maximum likelihood estimation without the priors, I get extremely wide confidence intervals. It's hard to tell anything. Again, those are neutral priors, but they're not non-informative. I'm saying I want to disallow things that I just think are biologically unrealistic. Sampling over the tree blocks really actually does help. It doesn't shift things very much, but if you're interested in whether a confidence interval overlaps 0 or not, that actually pushes it up enough that we can get into the scientific literature with all the magic cutoffs we need. The gain and loss priors turn out not to matter very much. Maybe that's also partly because we know so little about loss of accessory glands that ratio isn't really doing very much. And the full 20, if I allow everything to vary, which really biologically it should, because really whether you have accessory glands or not might also affect whether you gain male parental care or not, that simplification to only 12 parameters was actually important in allowing us to see anything in this tiny, noisy data set. I told you this took, so I think I've got a couple minutes left. Yep, good. Talking really fast. Not necessarily good. So the maximum likelihood optimization, that's the first thing I showed you, where we put this into this existing software package and let it find the best fit of the model. We've got, so these were some simulations I was running. And I was running things in parallel with a phylogeny with 500 tips and 1,000 edges. So it's a balanced phylogeny, so there's twice as many edges as tips, and 12 rate parameters. And so this whole thing took 12 to 24, this whole MCMC thing took 12 to 24 hours. A single fit took about 3 seconds with my new computational machinery. The new computational machinery is using, so there's going to be 1 to 3 hands. Automatic differentiation. OK, I'm in the range. There's 1. Automatic differentiation is another really cool computational trick, which allows you, when I'm doing these kinds of problems, I'm typically trying to go to optimize something, to go downhill or uphill. And so if I have information on not just the goodness of fit, but the derivative, so the effect of a change in the parameters on the goodness of fit, that allows me to optimize this thing much better. And I'm going to, oh yes, this is definitely an old version of the slide. The bottom line is I can speed this whole I can do the same calculation and speed it up by a factor of about 10 by recoding it with this automatic differentiation. And if you like this kind of thing, there's a package that you should look at, which basically allows you to write your model in R, but then automatically do all of this magic computation really quickly. That I'm working on with the core HMM, with the package owners, on integrating this into their package, so that if you were using their package before, you can now basically do the same problem you were doing 10 times faster without doing any work yourself. The other thing that I'm working to get into that package is a system for specifying the contrasts that I want to fit. So specifying the model that I ended up doing was, let's assume that the rates of change of parental care and spawning mode are independent, and that the rates of gain and loss of accessory glands depend on the other states. I want to be able to write that out with a formula like I would in R. So I want to say PC tilde 1. OK, that means that parental care is changing at the same rate regardless of what the other traits are doing. We're pretty close to having that working, which is exciting for me. What else? Yeah, there's more technical things that I'll talk about if people are interested. I guess I'll leave that up. Nope. I will leave that up in case people want to, in case those words mean something to somebody and they want to ask me to talk about them.
Have quite a bit of time, so feel free to ask anything you want to know about what Ben's talking about. Anything and everything.
Yeah.
Connected
Other topics are OK too. I don't know much about sports, so don't ask me about, and please don't ask me about politics. Yes.
I have a question about information for some species.
Yes.
Is there any way for the stats whether the missing as of information in the block of biology take mind the results somehow?
That's a really good question. I want to use your question to leverage something to say. I will answer your question in a moment. But I also want to say that one of the really nice things about the pruning algorithm. Oh, stop it. one of the really nice things about the pruning algorithm, if I can get back there, is that if I don't know what state if I know in the 8-state example, so if I'm looking at all 3 of these traits and I don't know the parental care, I can actually spread that probability. If I know that this thing is either in state 1 or 2, I can put a 0.5 and a 0.5 here. I don't have to put a 0 and a 1 and a 0. I don't have to be certain about the states at the tip, which was just very, very nice because it allowed us to use all of the data from those partially informed fish species without doing something really complicated. It could, the presence or absence of, sorry, the differential or informative presence or absence around that tree could definitely, I mean, I didn't think about it before 15 seconds ago, but it obviously could be a problem if the missingness was concentrated. That, not much of an answer, but basically yes.
Question from online.
Wants to know about RTMB and HMC.
Yeah, there's TMB stan, which you use nuts.
Yes.
Is that what you're using?
Yes. So so this is going to be letter soup to some people. I'm going to answer more of the question. TMB stands for Template Model Builder. And it's a system that some very smart Danish fisheries scientists have written that basically you write your likelihood or your probabilities in C++ code, and it compiles and it evaluates that very fast, and it also does automatic differentiation. So it gives you back a function that you can use in R that also comes with a function where you can evaluate the derivatives for those values. RTMB, okay, that's great if I'm willing to learn enough C++ to code my model. That's a big barrier. RTMB basically lets you write your model in R and kind of magically translate it into TMB so that you can write, again, you can write all of the code for this later thing is written in R code that you could look at and understand. Okay, I'm filling in the elements of this matrix. Now I'm looping over and doing this computation. Hamiltonian Monte Carlo is a form of Markov chain Monte Carlo that uses the gradients to move around to sample the surface in a more efficient way. There's lots of pieces here, I apologize. And TMB stan, and the stan is another software tool that people have written that does Hamiltonian Monte Carlo, but you also have to write your objective function or your posterior in C++. TMB knows how to talk to stan, so now RTMB knows how to talk to stan. So now I can write my model in R and then use Stan's algorithms to do Hamiltonian Monte Carlo. Again, I think the main audience for all of those words is not physically present, but hopefully it was useful to some of the other people online. Yeah.
So you have this relationship between parental care and the accessory gland
Yep.
from your complex model.
Yep.
If you just looked at the tree, could you see that there prior, or?
That's another really good question. That's another thing I haven't tried to do. I mean, this is the picture you would need to answer the question.
We have zeros and ones. Could we just plot it? Would you see that relationship without doing this complex modeling?
If I wanted to just do pairs plots or associations, the tricky part is it would be hard to dissect out the evolutionary part. I could do a chi-square test. If everything were independent, I could just say, how many of these guys with accessory glands have these combinations? How many don't? I would be done in 5 minutes. And if I chose to just lump everything into these 3 clades, then I'd only have 3 data points. And so I think it's hard to dissect out, oh, well, here's a clade where there's a little gray blob in here where these most gobies have accessory glands, but these particular gobies don't. So I think sort of dissecting all of that information would be tricky. I think it's a good idea, but it's a little daunting to incorporate the evolutionary history and the presence absence. Yes.
So either biologist or statistician, I'm kind of interested in these outlier points, and like the ones where you have like a red.
Oh, like these 2 breams that have accessory glands. Yeah. I need to call Segal back.
So as a biologist or a statistician, that's
I'm not enough of a fish biologist to have any idea why there's 2 bream species that have accessory glands. And probably, I mean, I would be guessing anyway. I would be taking, well, here's what's weird about the natural history of these 2 particular species, and maybe that's why they have accessory glands. No?
I never thought about this before, but like the height on a phylogenetic tree like this, you kind of mentioned as time.
Yes.
Would it make sense to, instead of looking at things on time, but looking at things in steps? Like, for example, that split right there, would that make more sense to compare that point to these other points where this, like,
So you could, there are various, and actually a lot of the older approaches to this didn't think about branch length. They just said, here's the ancestor of the ancestor. And we still do that sometimes when we don't have very much, when we don't have genetic data, if we only have taxonomic data, if we know these are the species, these are the genera, these are the families, then we could put something where basically each taxonomic level was 1 tree step up. You actually lose a lot of information if you don't have the branch lengths, because here's a really long branch, and I should expect a lot of evolution to have happened along that branch. Whereas if I collapse everything, I will think that that's the same as one of these branches. So in general, there's for the evolutionary biologist, or for, I guess, the computational statistical evolutionary biologists, all half of you in the audience, the other big assumption I've made is a Brownian motion evolution model. So I've assumed that evolution is basically just a random walk, whereas there are more sophisticated models like Ornstein-Uhlenbeck, which is sounds very fancy, but it says essentially that rather than species evolving at random, there's some kind of phenotypic or evolutionary target, and they're not evolving very far away from that target. You could, in principle, incorporate those things. Lots of people use the Brownian motion assumption because it makes everything easier. It's actually almost identical to making assumptions, Gaussian assumptions, normality assumptions in other contexts. Flip a coin.
So, I mean, I get why you have to treat these kinds of traits as discrete in this kind of large meta-analysis, but I'm wondering what is lost when that's done. Like there's underlying continuous variation in these traits, and like you'd expect, like a male carrier is important. You expect a fish with the most male carrier, like seahorses or pipefish, to have these accessory glands, but I don't see them on pretty.
I don't think, yeah. Well, that could just be because nobody's ever dissected the reproductive tract of a male seahorse carefully enough. Also, yeah, anyway, I don't know what their spawning mode is either, but I mean, the answer is yes. That biological variation is there. We've all, we talked about internal fertilization, pair spawning, alternative reproductive tactics where there are male sneakers. There's lots of biology that we have squashed out. and having squashed all of that complexity out, we have barely enough information to answer the question we were interested in, but we've certainly lost nuance.
This what I'm getting at is whether you, if you treated these traits as continuous, if you had data on the magnitude of male care with some relative investment accessible, would that enable improvement of your model?
Yes, yes, but I think it would make collecting the data, which is both what did the original scientists who were studying the species do, and then can we actually retrieve that information from the literature in a consistent way, way harder. But yes, I would rather continuous traits have way more information in them than just yes or no. And that should help. Oh, okay. There's several, I don't know whether those are old chat messages or. Okay.
Any other questions for Ben?
Yeah.
That you came up with a loss gain ratio.
Yep.
Did you just pick something that felt realistic, or did you look at the literature? Like, how did
I asked my colleagues, my fish biologist colleagues, what do you think is a reasonable range of asymmetry for the probability of gaining or losing? And we picked pretty broad rates, pretty broad ranges. So they were biased towards gaining, but we didn't disallow the possibility. They weren't very strongly biased. And they were, I guess Bayesian priors are the old vibes, or vibes are the new Bayesian priors or something. Yes. Oh, it came back.
Based on what you've done here, are you able to point the mechanism of change for the fish to either develop or not develop?
Absolutely not.
But do you think if we're able to pinpoint like a commonality between, I guess, the different, have the catfish, and what
Right.
the different red groupings, like,
Right.
do you think there's a commonality between those specific fish? I mean, based on what they even look like visually, they look similar.
I mean, if somebody wanted me to do that, I'm not sure this is answering your question. If somebody wanted me to do that and gave me a whole bunch of money, I would do a whole bunch of genomics and study the kind of genetic basis of these traits and try to figure out, okay, are they all adapting pre-existing pathways in the same way? So I think again, given the sparsity, I think narrowing down a lot about the ecological context would be very hard. Yeah.
So, of this whole project,
Yeah.
what do you think is the one piece that's going to go beyond this project and be a useful tool for other?
Oh, no, I absolutely, I mean, people do this kind of thing a fair amount. The Bayes traits package, which is the one I don't like, is very widely used. The Core HMM package was developed actually originally to fit something called the covariant model, which basically imagines that evolution is happening differently in different parts of the tree. And so you can have transitions among the traits, but you can also have transitions among regimes. You can say, oh, at this point, it became much easier for these traits to gain and get height.
Yeah, class.
You told me we had it for 2 hours.
Hmm.
Sarah, we'll clear out. Give us 5 minutes.
Okay, all right.
So I will shorten that. I think the ability to fit these models faster and to do actual Bayesian sampling, and especially this is the philosophical point, to move away from are these two rates equal to asking easily how different are these two rates and what's my uncertainty. I hope will actually, and I hope that by building it into a package that people are already using, that will propagate.